Hello everybody. Today's session is the question answer session. Today we will be discussing various questions on amino acids, proteins and nucleic acids. These questions have appeared in various competitive exams. So let's start. The first one is, which purine base is found in RNA? This is a common question and we all know the answer. Answer is uracil. In RNA, thymine is replaced by uracil, which is the demethylated form of thymine. Next, which is the most abundant protein in the animal world? Trypsin, hemoglobin, collagen and insulin. The answer is collagen. Collagen is a fibrous protein and it provides structural support to the extracellular space of our connective tissues. It is very rigid and resistant to stretching. Next one is, which one has a tertiary structure? Insulin, mRNA, ATP or rRNA? Insulin has 3 disulfide bonds in its molecule and it has no tertiary structure. mRNA has no tertiary structure. It has some few loops and some helices. ATP, adenosine triphosphate. It is not a polymeric chain. rRNA has the tertiary form. Next is, which one is not a polymer? Lipid, RNA, polysaccharide or myoglobin? The answer is lipid. And why? We know RNA is ribonucleic acid, that is the polymer of nucleotides. Here Mars are nucleotides. We know polysaccharides and myoglobin. Myoglobin stores oxygen in muscle tissues. It is a protein with long chain of amino acids. Lipids are not considered to be polymers because lipids don't contain the repetition of mass. Polymers means repetition of monomers. The basic units of lipids are fatty acids and glycerol. They don't repeat in the chain. The reason for double helical structure of DNA is operation of hydrogen bonding, covalent bonding, van der Waals force or disulfide bonding. Covalent bonding, we know phosphodiester bonding, that is in the backbone chain of DNA. And in between purine and pyrimidine there is hydrogen bonding. So the answer is A. 1 turn of the helix in a B form DNA is approximately, these are the values. Answer will be 3.4 nanometer. For details, you can see the video on DNA facts. Which of the following glucose transporter is insulin dependent? GLUT4, GLUT1, GLUT2 or GLUT3? We have to know some few points about all these types of GLUTs. GLUT1, it is present in almost all cells. GLUT4, it is in liver, also in beta cells, small intestine, etc. GLUT3 we can find in neurons, placenta, etc. and GLUT4 in cardiac and skeletal muscles. GLUT1, GLUT2 and GLUT3, they are insulin independent, but GLUT4 is insulin dependent. When glucose is more in our bloodstream, then insulin becomes active and in that way GLUT4 becomes active. There is GLUT5 also and helps in transporting fructose. Next, Rubisco is the abbreviated form of ribose biphosphate carboxylase oxygenase or ribulose 1,5-bisphosphate carboxylase oxygenase. The answer is B. We know Rubisco is the most important enzyme in the world. It is produced by all green plants and responsible for fixing carbon in the form of carbon dioxide. Where it is found? It is found in the stroma of all chloroplasts. Next is ribose and deoxyribose can be differentiated by Fehling solution, Tholin's reagent, Osazone formation and Barford's reagent. And the answer is Osazone formation. Ribose and polyhydrazine. They both react and form osazone. Because of this 2' OH group which is not there in deoxyribose, polyhydrazine can react with ribose. Amino acids are mostly synthesized from mineral salt, fatty acid, volatile acid and alpha-ketogluteric acid. The answer is alpha-ketogluteric acid. Now to explain this, I have to show you the path of glycolysis and citric acid cycle. I have drawn a simple sketch starting from glucose. After glucose it is G6P, that is glucose 6-phosphate. From here we get histidine through various steps. Then it is 3PG, that means 3-phosphoglycerate. This is serine. And this is E4P, that is erythrose 4-phosphate. And PEP, that is phosphoenol pyruvate. So erythrose 4-phosphate and phosphoenol pyruvate, they give you F, Y and W. Do you remember the names? I'll tell you. F is phenylalanine, Y is tyrosine, W is tryptophan. Then come to CAC, that is citric acid cycle. So from pyruvate, then citrate, then alpha-ketoglutarate. This alpha-ketoglutarate gives us Q, P and R. Q is glutamine, P proline, R is arginine. Then it is oxaloacetate. Oxaloacetate gives us N, M, K and T. That is asparagine. N is, we all know, methionine. Then K is lysine and T is threonine. So you understood the source of all types of amino acids in our body. Main is glucose. Next comes, in an alkaline medium, glycine predominantly exists as A or N, cation, anion, Zwitter ion and covalent form. This is a very interesting question. This C is the chiral center that you remember. But in case of glycine, these two will be H. So in glycine, there is no chiral center. And CO minus and NH3 plus. In alkaline medium, this OH minus will take up one hydrogen ion from here. And this is the format. So this is an anion. And in acid medium, what will happen? That H plus will come over here. And then cation will be the answer. What are the most diverse molecules in the cell? Answer is protein. We all know this. Which one correctly represents the structural formula of the basic amino acid? In amino acid, we know there is NH2 and COH groups, but one NH2 and one COH. So this A you have to discard. There NH2 and CH2OH, no COH. So discard this. NH2, COH, another NH2. Discard this. Which is not consistent? Important. Not consistent. Which is not consistent with the double helical structure of DNA. A equals to T, C equals to G. That we know. That is Chargaff rule. density of DNA decreases on heating. When you heat the double helical structure of DNA molecule, it breaks down. The hydrogen bonds between the purine and pyrimidines, they break down and you get two separate chains. It depends on the temperature of the heating and the density of DNA decreases. A plus T and C plus G ratio is not constant. In a species, A plus T and C plus G ratio is always constant. So this is correct, this one is correct, this is constant. So not constant is not consistent. So this is the answer. Which one is the correct polypeptide example of an eukaryotic cell? A, U, G. This is the start codon. And this start codon always goes for methionine. The answer is A. That we have seen in the video of mRNA. In bDNA, the number of nucleotides per turn is 11, 10, 15 or 12. The answer is 10. 11 is for aDNA and 12 for zDNA. A segment of DNA has 120 adenine and 120 cytosine bases. The total number of nucleotides present in the segment is 120, 240, 60 or 480. Let us see. In a DNA double helix, as per Chargaff's rule, we know A equals to T and C equals to G. A is 120 here. So T will also be 120. C is 120, so G will also be 120. So the total number of nucleotides, they are these, all four, that is 480. Is it clear? Antibiotics bind to small subunits of ribosomes of prokaryotes and they attack their cell walls, inhibit protein synthesis by their ribosomes, attack the rRNA of prokaryotes and change their genetic code. They inhibit protein synthesis and B is the answer. Our next question answer session will be on enzymes. Thank you for watching.